My name is Angelin Beischlein, and I am a senior studying medical laboratory sciences. My project investigates the mechanisms underlying thrombocytopenia in malaria. Malaria is a serious disease caused by the parasite Plasmodium, which infects red blood cells. In 2020, there were 241 million cases and 627,000 deaths caused by malaria. Sub-Saharan Africa carries 95% of malaria cases and 96% of malaria deaths. About 80% of these deaths are children under 5. Thrombocytopenia is a drop in the circulating platelets and a frequent symptom of malaria. The mechanisms underlying thrombocytopenia in malaria are unknown. While thrombocytopenia isn't a severe complication in regards to bleeding issues, it is a sign of poor prognosis of malaria. More research is needed to determine the underlying mechanisms of thrombocytopenia in malaria infection. We use mouse models of malaria to study these mechanisms since thrombocytopenia is also well established in these models. This data from the mouse models Plasmodium chaboti and Plasmodium uelii show that as parasitemia increases, platelet levels decrease. As parasitemia decreases, platelet levels return to normal. Possible reasons for thrombocytopenia in malaria are 1. The consumption of platelets, and 2. The lack of production of platelets. This data suggests that both mechanisms are at play. First, we investigated the mechanism of platelet consumption. When platelets are consumed, they are adhering to the endothelium, or the inner lining of the blood vessels. This would cause a drop in the number of platelets circulating through the body. I conducted an experiment using genetically altered mice that allowed us to deplete their megakaryocytes through diphtheria toxin injections. Megakaryocytes are cells in the bone marrow that produce platelets. Then, the mice were infected with Plasmodium chaboti. The megakaryocyte-depleted mice then had platelets transferred into them. While their megakaryocytes were depleted, platelet production was removed as a factor. This means that if platelet levels still decreased over the course of infection, it would be due to platelet consumption. In this experiment, these platelet-depleted mice still experienced a drop in platelets. This data supports the hypothesis that thrombocytopenia is caused, at least in part, by consumption of platelets. The next section of my poster investigates the question, what are the implications of platelet consumption in malarial pathogenesis? Platelets may mediate parasite sequestration in organs such as the spleen, preventing the parasites from being cleared from the body. This data shows that infected mice with defective platelets, called NBL mice, actually clear the parasites faster and have lower overall levels of parasitemia than infected wild-type mice. NBL mice have platelets that don't activate or perform their function correctly. Our hypothesis is that if the platelets can't activate, then they can't mediate this parasite sequestration, and the parasites will be cleared from the body faster. This is an interesting direction for future research. Next, we investigated the possibility of lack of platelet production. Data previously collected in the lamb shows that megakaryocytes decrease in number during infection. To further explore this, we infected mice with Plasmodium chaboti and extracted their bone marrow. Then I performed flow cytometry to measure markers of megakaryocyte and megakaryocyte progenitor cell apoptosis. This preliminary data suggests that this drop in megakaryocytes could be because the megakaryocyte progenitor cells undergo apoptosis before they can become mature enough to produce platelets. However, we still don't know why the megakaryocytes or their progenitor cells might undergo apoptosis during infection. The next section of my poster investigates the question, could it be related to bone marrow inflammation? Data collected previously in the lamb lab through RNA sequencing shows that a cytokine associated with inflammation, called interferon gamma, is upregulated in the bone marrow during the course of infection. The interferon gamma receptors are also upregulated, showing that interferon gamma is present and able to perform its inflammatory function during infection. To investigate this further, I infected genetically altered mice referred to as great mice with Plasmodium chaboti. These mice express interferon gamma fluorescently. making our flow cytometry measurements more accurate than traditional antibody staining methods. After infection, we extracted their bone marrow and used flow cytometry to measure interferon gamma expression. I found that there was both a higher number and percentage of interferon gamma expressing cells in the infected mice compared to the uninfected mice. This data establishes that the bone marrow is inflamed during malarial infection. Further research will investigate the relationship between the drop in megakaryocytes and this bone marrow inflammation. Thank you for listening, and I would be happy to answer any questions about this project. Thank you to the Access Program, the Office of Undergraduate Research, and U of U Health for funding this research.